Thank you for being here. It's a very exciting occasion. I'm Phil Duxbury. I'm the Dean of the College of Natural Science, and I'll be your MC for today. This is a joyous occasion for the Department of Chemistry, for the college, and the university, as we honor and celebrate Dr. Tuo Wong as the inaugural Carl H. Brubaker Jr. Endowed Professor. It's great to have you with us today to celebrate this. Before we go too far into the formal part of our program, I am delighted to share that the donor of this prestigious endowed professorship, Professor Jim Heshley, is here with us this afternoon. Jim, thanks for being here and for all that you have contributed and continue to contribute to Michigan State University. Thank you. As we recognize Dr. Wong for this prestigious honor, we're looking forward to hearing from Professor Heshley later. I'd also like to welcome Jim's daughter, Heidi, and granddaughter, Olivia, who I'm told is actually just come from class, a Michigan State student, which is doubly great. And of course, a big welcome and thanks to Tuo's family members, colleagues, and lab members who are here today. Our program today will touch on the importance of the Carl H. Brubaker Jr. Endowed Professor designation and the long-term impact that endowed faculty positions have on the continued excellence of the university environment. Let me personally congratulate Dr. Wong for his outstanding achievements to date, which are recognized by this prestigious endowed professorship. Following a few remarks and a brief introduction about Dr. Wong from Chemistry Chair Tim Warren, we will hear from our honoree and have his formal investiture. Finally, Professor Heshley will provide a few remarks about how he came to establish the Carl H. Brubaker Jr. Endowed Professorship that we are conferring for the first time today. With that, it is a great pleasure to turn the program over to MSU Interim President Teresa K. Woodruff, who will share remarks on behalf of the university.
Well, thank you, Dean Duxbury, and it's always a pleasure to be part of the College of Natural Sciences and the honorifics that you bestow. And to the people of chemistry, welcome. It's good to see all of you here. And I'm particularly delighted to be here to recognize Dr. Tuo Wong on your installation as the inaugural Carl H. Brubaker Jr. Endowed Professorship. I also want to welcome your family members, your distinguished wife as well, a great faculty member at Michigan State, and an upcoming faculty member at Michigan State University, young Felicity. So Felicity, I look forward to seeing you here as a faculty in the future. So to you and your family. And Jim Heshley, Jim, thank you so much for all that you have done. Jim is a 1965 chemistry alumnus, as many of you know, and generously endowed this professorship in the name of his mentor. And your impact on MSU is long and wide, and particularly, and I'm sure there will be statements made later on cisplatin and particularly carboplatin, and your work in that process of bringing those cancer drugs to market and to the public. And on behalf of a grateful university, thank you for all you've done, both in the macro and today for Tuo. We also welcome your family members as well. And Michigan State University family is a family. And so we're delighted that as we are gathering together as Spartans, we have family members that are doing this as well. And it is really in that spirit that I congratulate the college itself and Dean Duxbury for your leadership in the way that you enable this group of chemists to come together in a way that they wouldn't otherwise. These investitures are important because they provide funding that allows you to do the unexpected, to allow you to be creative, to allow you to grow and to nurture, and to have the greatest kind of success that we expect of all of our faculty. But with these with this endowed professorship, we hope that it gives you yet another platform on which you can see over horizons and imagine taking your work in different directions. Dr. Wong, I did a little research on you, and that's kind of what I do. And I've been impressed by the work that you've done in solid-state nuclear magnetic resonance technology, in particular work at the nanoscale of the fungal cell wall. Some of those papers are really cool in terms of the novel antifungal therapeutics, and I think that the world desperately needs new ways to combat fungi and microbial infections, and I expect that this work is going to have magnified effects around the globe. I was also delighted that you were an NSF early career awardee, and that's a great milestone for junior faculty, but I call it a touchstone. It's not the place you get to, it's the place you touch and move on from. And I'm really pleased with that and some of your work in training the next generation, particularly from kindergarten all the way through postdocs, to broaden the training for biophysics and biomolecular studies, making it more accessible to all. That is, friends, what we are all called to do, and I'm really delighted that the way in which you have utilized those dollars to enable that broadening is really significant and really impressive. And so it has become my custom in these events to give a charge to the new honorand, Dean Duxbury, and you have sat through some of those, and I guess I should come back and see if they've worked, to see if, in fact, my charges have had any effect. But on this occasion, Dr. Wong, I'd like to give you a charge as you accept this award later in the ceremony. I believe that endowed professorships are among the highest honors a university can bestow. They're given in recognition of the work that has been done, but with great expectation for the work that will be done. And so I charge you to not merely hold a title, but to use the title and the confidence we have in you to untether yourself from conventional thinking, to create impact that benefits the nature of an endowed professorship and our investment and trust in you. This is an award, not a reward, a name in anticipation of work, not work that is in the rearview mirror. And so today, I want to thank very particularly the vision of Jim and his respect for his mentor in naming this the Brubaker Endowed Professorship. I want to celebrate with all of you in the Department of Chemistry the conferral of this new award. I also want to recognize the excellence of the environment in which this work is done. Work at Michigan State is never done in isolation. It is done within the context of the college and the department. And I also want to make sure that we think of this not just individually, but in the context of the upward trajectory of Michigan State University itself. It is in that interest that we serve, and it is in that way that I bring congratulations to you on this occasion. Congratulations.
Thank you, Interim President Woodruff. Again, Tuo, on behalf of the College of Natural Science, congratulations on this well-deserved honor. As Professor and President Woodruff just said it, named faculty chairs and professorships are very important both for the individual, the college, and the university. To be competitive in this current world of academic life, we need endowed positions. We actually need them. We need more of them to attract and retain the best. So this is a great honor to receive this from the Heschle family, and we're looking forward to the great progress that Tuo will make. The other thing I want to say is that I really am very, very pleased to have you here for another reason. We had a big investment in NMR technology about 4 years before you got here, and we were committed to hiring top-notch people to help us utilize those facilities. And Doug Gage, who is here, our Vice President for Research, made a big investment in that as well as us. So I've been worried about that for the last 3 or 4 years, about whether it would pay off or not, and today I feel great, The bestowing of this professorship to Dr. Wong will help guarantee Michigan State's continued capacity to enhance its national and international reputation in the development and application of solid-state nuclear magnetic resonance technology and applications. The impact of these types of endowed positions is much broader, though. When you have them, you can attract stronger students and other faculty to Michigan State and to the Department of Chemistry. So now we have within NMR an area of focus and excellence in this area, which we've had before, but not to this extent. So this is really a step up for us in this area. All of these aspects help ensure that our future alumni will have the education and experience they need to compete for the best jobs and to lead the nation and world in maintaining and building scientific leadership. To Woe, no pressure, but we're looking forward to great things. With that, I'd like to turn the floor over to Dr. Tim Warren, Chair of the Department of Chemistry, to say a few words on behalf of the department and to introduce Tuo.
Thanks, Phil. This is really quite a special occasion. I'm really happy to be here with everyone to celebrate the investiture of Tuo Wong as the inaugural Carl H. Brubaker Jr. Endowed Professor. So before I introduce Tuo and his work, I want to share a heartfelt thanks to Dr. Jim Heschle for making this opportunity possible. Jim is an alumnus and a dear friend of the chemistry department with whom we shared a special relationship now in its 7th decade. This endowed position was absolutely critical in attracting Tuo to Michigan State. While we were trying to recruit him, he had a competing offer with also another endowed professorship at an institution that was also dear to his heart. Not only did Jim's gift bring Tuo and his outstanding scholarship to Michigan State, but it also enabled us to hire Professor Weiwei Xi, his spouse, whose career is also on a meteoric rise. And now a few words about Tuo. He received his bachelor's degree in chemistry from Nankai University in Tianjin, China, and started his PhD in physical chemistry at Iowa State University with Dr. Mei Hong. He then moved with Dr. Hong to Massachusetts Institute of Technology, where he completed his PhD in 2016. After a year of postdoctoral research there, he started his independent career at Louisiana State University in 2017. Employing nuclear magnetic resonance spectroscopy, or NMR spectroscopy, as a valuable tool to investigate the structure and dynamics in biologically important molecules, Tuo and his team study the carbohydrate-rich cell walls of plants and fungi. These serve as both a central source of biorenewable energy as well as a natural target of antifungal agents, but their structure turns out to be very difficult to study on the atomic scale due to lots of different factors: insolubility, disorder, and just the inherent complexity of these biological polymers that make up the cell wall. Advanced NMR methods in his laboratory that include dynamic nuclear polarization, or DNP, enable Tuo and his team to uncover invaluable molecular insights into the structural variation and complex motions of biomolecules. These findings deepen, revise, and advance our understanding of the 3D architecture of these complex biological materials, which may guide the engineering of better crops for higher biofuel yields, as well as the next generation of antifungals. Tuo's research career is on a steep ascent. In 2020, he received the NSF Career Award, as well as the DOE Early Career Award, each foreshadowing his emergence as a leader in the area of solid-state NMR for biological materials. Soon thereafter followed the Anatole Abragin Prize for the International Society of Magnetic Resonance for pioneering applications of solid-state NMR in dynamic nuclear polarization. Attracted to Michigan State by our outstanding nuclear magnetic resonance facility that Phil has made mention to, and a world-renowned plant science community that MSU is known globally for, we were really lucky to recruit Tuo in 2022. Since his arrival, he's added to his funding portfolio a major NIH grant to study fungal pathogens and as well as antifungal agents. On further ascent in the NMR solid-state community, he received the 2023 Varian Young Investigator Award in Magnetic Resonance. Tuo's research group is also very active in the community. Last year, his team of 3 postdocs, 8 grad students, and an undergraduate presented 6 talks and 14 posters around the U.S. Please join me in welcoming Tuo Wang, who will now share with us his fascinating science.
Okay, thank you so much. So it's such an honor to be here. I really would like to thank everyone for your attendance. And also I would like to give special thanks to our respected visitors, our president, our vice president, Dean Duxbury, and my chair, Dr. Tim Warren, who is helping me throughout everything. So as you can see that my research is focused on NMR spectroscopy, and we use the NMR technique to investigate many different carbohydrate polymers in living organisms. And adding a new dimension to this is this endowed professorship, which has been made possible by Dr. Jim Hochschild, and also with his unreserved support. So today I will only give you a brief flavor of my career and also my research. So I always have this question from audience. So what kind of research are you doing? So every time I will say it very proudly, I'm an NMR spectroscopist. Every time I have the same response, could you speak English? Yeah. More broadly defined, I'm actually a biophysical chemist, and our research is on the interface of physics, biology, and chemistry. More specifically, we're using some really simple physics techniques that can even be handled by a 5-year-old kid. It's called NMR, to characterize the beautiful structural carbohydrate components to understand the important biological questions related to living organisms such as plants, fungi, and bacteria. So NMR is a technology started from physics. And in the early stage, we have the Nobel Prize related to magnetic moment and resonance measurement. And later, we have the high-resolution NMR spectroscopy and NMR structure biology. And more recently, we have the development of magnetic resonance imaging, which is widely applied in hospitals. Because we have a large collection of NMR active nuclei in nature, we have versatile capabilities. For example, we can use proton and fluorine to characterize the structure of organic compounds, pharmaceutical compounds, as well as proteins. And we have phosphorus that allows us to look at the structure of lipids in membranes, as well as nucleic acids. And we also have lithium, which allows us to look at the battery and think about how it's being charged and discharged. In my lab, we mainly use carbon-13 and nitrogen-15 together with a little bit of proton to handle the structure of carbohydrates. In my PhD and postdoc research, my research is heavily focused on protein assembly systems, which are crucial to viral infection and neuron disorder disease. And this is a fast lane of research with a lot of investment from funding agencies such as NIH, and you can easily publish better papers there. But when I started my independent career in 2017, I asked myself a question. How could I distinguish myself from my fellow researchers on the same track? And how could I make a unique contribution to science? And at that moment, I got fascinated by the beautiful structures of complex carbohydrates, which are also a major family of biomolecules, but substantially under-investigated. They're important to many biological processes, such as energy storage and cellular recognition. And I decided to take this risky path, and I will feel the untouched beauty in this underexplored territory of science. Fortunately, after 5 years of struggling and a lot of failures, we were able to establish a comprehensive research program focused on carbohydrate NMR with biomedical relevance and bioenergy and biopolymer production, as well as the methodology development. On the biomedical part, we are looking at the carbohydrate armor that protect the microbes from being targeted by antifungal treatment. And in the energy part, we are looking at the interface between the lignin, which is aromatic polymer, and the carbohydrate components such as cellulose and hemicellulose, because this is a major interaction that has limited our use of biomass for energy. And we also started the microalgae research by looking at the starch, salt wall polymers, as well as the glycolipids, and that's a recently started project. We only have one publication followed by another one that will show up soon without funding yet, and we are seeking for funding for this. And my lab also have dedicated efforts on methodology development, and this is outlined by two streamlines of projects. First, we want to make NMR more accessible to allow the rapid screening of natural samples without labeling. And second, we are coding a solid-state NMR database of carbohydrates to facilitate the sugar analysis to benefit the community. So today, I will only use a few slides to give a brief flavor of some of the projects. So first, we'll talk about fungi. So why fungal infection even matters? In fantasy, it's described a lot. For example, in the recent TV series and also the very famous game under the same name, The Last of Us, the zombies are depicted to be our enemies, and those zombies are infected and controlled by fungi. And the producer said to the interviewers that, okay, this concept is inspired by the behavior of fungi in nature because they can infect and control insects and finally digest them for nutrition. And there's a very bold claim at the beginning of this TV series. What if, for instance, the world was slightly warmer, and now there's a reason for the fungus to evolve, and we will lose the fight against them? While I really appreciate this claim, I want to mention that the producer really didn't do a great homework. Because in reality, fungal infection is already a serious problem. And in the United States alone, the annual inpatient costs associated with treating the major invasive species of fungi is 4.6 billion and is increasing. And we have more than 2 million patients suffering from invasive fungal infections worldwide every year, and then the mortality is very high, 20 to 95%, even after treatment. So those infections that can happen to the brain, to the lungs, to the kidney, and here I will take Aspergillus as an example, which is a major prevalent airborne fungus. Underground, we have the mycelia formed by hyphae network, which is responsible for the vegetative growth of the fungus, and we also have the conidia. Every moment, we are breathing in the spores, but if we have some immune deficiency, the spores will invade to the lungs and develop mycelia again, similar to those developed in soil, but now happening in the lungs. And we have very limited choice of antifungal compounds, and most of them are very ineffective. And the recent efforts have been devoted to develop antifungal agents targeting about salt wall carbohydrates, because they are unique. They are not present in our cells. They are safe target. So my lab wanted to help this process by providing structural information with the fungal cell wall and its remodeling mechanism during the antifungal treatment. And this is done by first carbon-13, nitrogen-15 labeled fungi, and we can directly pack the living cells into a small container without any treatment, and the fungi is alive during our measurement. And we can measure a lot of high-resolution NMR spectroscopy, which I will skip here, and we can get the local structural difference, intermolecular packing, water retention, dynamics, as well as the domain distribution of tons of different types of polymers within the living cell, and get the structural information and to give the model. And I will skip the streamline of the studies done by my lab in this, on this topic and directly lead you to this cartoon illustration of the fungal cell wall. So outside the membrane, we have a carbohydrate called chitin and another one called alpha-glucan, tightly associated to form very rigid, waterproof cores. And then those cores are distributed and supported by a hydrated and soft matrix formed by diverse linked beta-glucans and further protected and covered by another layer rich in glycoproteins. So now the next question we want to answer is how this armor, carbohydrate armor, will be rebuilt by the fungus when we give it antifungal treatment. So the antifungal drug we are using is caspofungin, which has already been clinically approved and used in treatment. And you can see that the drug-treated culture has substantially reduced growth compared to the non-treated culture. And the cell wall thickness becomes substantially thicker, increase from 120 nanometer on average all the way to 180 nanometer. And because this drug is targeting the protein machineries synthesizing beta-glucan for the cell wall, as expected, you can see all the blue signals labeling beta-1,3-glucan, they are missing in the NMR spectrum of the drug-treated sample. However, we have substantially higher content for the chitin molecule, and we have 2 new forms of alpha-glucan. So clearly, the fungus has reshuffled its biosynthesis to overproduce and produce new forms of other polysaccharides to compensate for the loss of beta-glucan caused by the drug treatment. Another major finding we found is the decrease in the water retention in the cell wall. The cell wall becomes more hydrophobic. So clearly, you can see that the fungus has fully rebuilt its carbohydrate armor with better protection compared to the previous version, and produce thicker and more hydrophobic and more rigid cell wall that can efficiently exclude the water-soluble antifungal drugs that we are using these days. It also gave another 2 clues for the antifungal strategies. First, we need to simultaneously inhibit the biosynthesis of multiple carbohydrate components instead of a single one. And second, we need to develop a better antifungal drug that can target the modified carbohydrate structures remaining in the fungal cell wall. And our research is basically informing us on how to give more effective armor-piercing attack to those invading microbes. So now I will change the flavor. I want to talk a little bit about the plant research because MSU is a paradise for plant research. We are one of the major institutions participating in the Great Lake Bioenergy Research Center, and we have the great plant research lab on campus. So my lab works on both the primary and the secondary cell wall, and they are found in the fast-growing part or the mature part of the plants. And in both, we can find many different types of carbohydrates. For example, the fibrils formed by cellulose, and we can have those strands of hemicellulose, such as xylan. And in secondary cell wall, we have the additional deposition of the aromatics called the lignin. Yet the interaction between the aromatics and the carbohydrates, they gave us a great issue because the carbohydrates are no longer that accessible to enzymes and chemicals, and we cannot easily release them for energy. So my lab wants to use NMR to give snapshots of those complex structures in order to guide our scientists to find better targets and conquer such kind of interaction. So here's the second spectrum that I will show today, out of 4 total. I tried my best to minimize the number. And you can see the beautiful spectrum measured on the intact stem of the eucalyptus, which is hardwood. And you can see the greenish texture and color of the sample in native state packed in a glassy NMR rotor. And here we can get atomic level resolution. For example, we can track down the carbon connectivity from carbon 3 to carbon 4 to carbon 5 for the 2-fold xylan, which has a flat ribbon structure in space. And we can track down the carbon connectivity with 3-fold xylan, which has a zigzag structure in space. And we can track many glucose units in cellulose, which has complexity locally in the conformation and higher order for the assembly. And we can track all the different carbon signals in different polysaccharides in the whole stem. And following that, I skip all the details, but will directly show you the cartoon illustration that details the interactions between the carbohydrates and the aromatics in different plant species. Now we look at the grass species because they include all the food crops we have, for example, rice, corn, and oat, and wheat. And we look at the hardwood, such as eucalyptus and poplar, and we look at the softwood, which includes spruce and pines. And you can find an increasing level of molecular mixing from grass to hardwood and softwood. And on the molecular level, we can distinguish different conformations of xylan with a zigzag conformation wrapping up the lignin nanodomains, which is a further bridge to the flat ribbon structure in red color coating the surface of cellulose microfibrils. In this way, this polysaccharide will bridge everything together and hold the cell wall together as an integrated part. And in the hardwood species, the chemical structure of the xylan has changed, but the principle of the zigzag and the flat ribbon structure still applies. and the lignin has been divided into smaller particles to enable better mixing with carbohydrate components. And in the softwood, we have the highest level of molecular complexity because of the introduction of another carbohydrate called mannan. However, all the molecules are well mixed on the molecular scale, and the lignin no longer exists as nanodomains. However, they now exist as a dispersive distribution to better engage with carbohydrate polymers. And I hope with those kind of structural snapshots of different plant species, people can understand those lignocellulose material better and can find the perfect target to release the sugar and without effect from the lignin components. All the structural studies conducted before are based on carbon-13 labeled materials, but we are also developing methods to rapidly screen unlabeled tissues and materials. For example, we grab a bit of soil from the ground and we can tell you what kind of carbohydrates are there and what kind of aromatics and proteins are there. Or we can take some red stem directly from the field without any labeling. Within 35 hours, we will be able to tell you all the carbohydrates and their different conformations, such as the flat ribbon structure and the non-flat structure with xylem. And this kind of capability is based on the technique called dynamic nuclear polarization, or DNP, which can shorten a lengthy experiment which previously required 1 to 6 years to finish, now in a single day. And here's a beautiful picture taken by my student Wan Chengzhao, who has perfectly split himself into two flash persons, doing experiments and analyzing data at the same time. Yeah. My lab also coded complex carbohydrate magnetic resonance database, CCMRD, which is publicly available to our community. So we use it as a platform to deposit data and use it as a platform to allow us to further develop automatic analysis tool to benefit our facility. So if now let's summarize my projects. If I think about it, the NMR spectroscopy is a foundation. And growing on top of it, we'll have different branches on plant, fungi, and algae. And what this professorship allows me to do is to integrate my research program better with the available expertise on the campus. For example, we have started collaboration with Josh, who is also here, to combine NMR experimental inputs with modeling power. And we develop a new project on the bacteria and antimicrobials with collaboration with NEO and the NEO's lab. And we even step into the application side by looking at the bioproducts by collaborating with Morgen. At the same time, we are carefully culturing two small seedlings based on totally different techniques. And the professorship here is basically the nutrition and the sunshine that allows us to counter risk, and hopefully they will grow up into two complete streamlines of projects in the future. It also pushed me to think forward, how I can benefit our community and our campus. So if we think about television and monitors, we know that we have technical evolution from the HD all the way to 4K Ultra HD these days with better resolution. The same evolution is ongoing also for NMR. Nowadays, we are no longer talking about 800 megahertz, 600 megahertz, but we are talking about 1 gigahertz, 1.2 gigahertz stage. In Europe, they are much ahead of US. They have built this facility network with 26 user facilities across the whole European Union and building gigahertz NMR capability for each of them. And in the United States, we have the first 1.5 home-built NMR instrument in Tallahassee MegaLab, which absorbs 1/4 of the electricity of the city Tallahassee. Whenever in operation, all the light will suddenly become dim. So this is not the way to go. It cannot be run for a long time. But we also have the 1.2 gigahertz NMR instruments funded for Wisconsin, for Ohio, for Georgia, and for Tennessee. And I hope with my efforts, we can become another major site on this national network with home base at MSU. Yeah, so I would like to thank the people, especially my group members who did all the research. My current group members are all here, and my visiting scholars, alumni, as well as undergraduate co-workers. You can see that our group has rebuilt a little bit. We have shrunk to 5 people in 2022 when I first relocated to East Lansing, and now we have fully rebuilt with MSU blood. And I would say that the people is the most fantastic part of the lab, and I always enjoy the moments working with them, celebrating with them, and watching them how to develop into independent researchers. And this is exactly what is done by Professor Brad Becker, who has supported many young scientists and students. And one example at that moment was Jim, who has later contributed a lot to the development of the gold standard antifungal drugs. And it's the people that has made the lab a great place. So I will stop here. I want to thank the instrumental support and also funding support from NIH, DOE, and NSF, and other funding agencies. I would like to thank my group members, and I want to thank my visitor, Jim Paul, who has traveled a long distance from France to join this ceremony. And finally, I want to thank my colleagues, co-workers, and collaborators for the support. I feel so honored to be here. I thank you so much for organizing this ceremony, and so thank you so much, Jim, for making this possible. Yeah, thank you.
Well, that was just an amazing talk. I even learned a lot. That was really good. So thanks, 12, for that. And now we have the official medaling. So we invite President Woodruff back to the stage for that.
This is the superpower part, so you get to clap.
We get to hear from the donor of this award, and so it's my pleasure to introduce the donor, Professor Jim Heshley, who will provide the closing remarks for today's program. Jim has had a remarkable career defined by both research and academic positions. He received his bachelor's degree in chemistry from Franklin and Marshall College in 1959, and his PhD in inorganic chemistry from Michigan State University in 1965. His first postdoc position was a rare opportunity to participate in the Trans-Uranium Element Program at Union Carbide and Oak Ridge National Laboratory. In 1969, Carl H. Brubaker alerted him to a job opening as a cancer research fellow in the laboratory of the famous Barnett Rosenberg in Michigan State's Department of Biophysics. Accepting that fellowship initiated a 50-year and counting research focus on the use of metal complexes in medicine and biology. Heshley's work with the Rosenberg lab focused on the synthesis and characterization of cisplatin, the anti-cancer drug for which Rosenberg is credited the determination of the in vivo properties of platinum radio-labeled platinum, chloroamine complexes, and synthesizing many cisplatin analogs, including the discovery of a very famous and important analog called carboplatin. Heshley is the co-inventor of that drug, and it was approved by the FDA in 1989. And it is now one of the major chemotherapy drugs along with cisplatin. Between 1972 and 1994, Jim held various positions in industry, pharmaceuticals, and academia, along with holding a teaching and research position from 1991 to 93 at the University of Michigan's Department of Chemistry. He returned to Michigan State as a chemistry assistant professor in 1994, where he taught general chemistry, a physical science course, and worked with undergraduate students doing research in the area of platinum cancer chemotherapy. He then joined Eastern Michigan University as an adjunct professor in 2009, where until recently he mentored undergraduate students in bioinorganic chemistry and occasionally introductory chemistry. He will soon be returning to Michigan State to work with some of the people here, and in particular in Tom O'Halloran's lab, which focuses on developing on quantitative understanding of the regulatory roles of transition metals in controlling cellular physiology. Jim's long-standing love of chemistry has saved countless lives and and positively impacted hundreds of young minds through his teaching, mentoring, and philanthropic support. Without further ado, it is my honor to invite Dr. Jim Heshley to the podium.
Thank you very much, Phil. I enjoy very much being here and hearing all the previous talks. We have quite an elite group, and we welcome Professor Tu Wong and his wife and daughter, and you have a very promising career here, to say the least. My PhD research program was with Dr. Brubaker in 1961 to 1966, basically. I had initially decided before coming to MSU that I was going to be an organic major. But because of the structure of the organic chemistry at that time, not having well organized prediction schemes, I switched into inorganic, fearing that I'd have to memorize all these individual reactions and statements. So anyhow, Dr. Carl H. Brubaker Jr., he's a very highly respected inorganic chemist, professor by professors and by students alike. He's an intelligent and effective leader who established good rapport with students, and needless to say, I respect him enormously. My undergraduate chemistry professor at FNM, as you heard, knew Dr. Brubaker and suggested that I attend graduate school at MSU and specifically work with Dr. Brubaker in inorganic chemistry. I took him up on the recommendation and indeed did both, attended MSU and worked with Carl Brubaker for those 5 or 6 years. Now, Dr. Brubaker gave me the opportunity to do a most unusual research project. It was a project relating to the photoacceleration of the electron transfer between uranium-4 and uranium-6 ions in solution using the rare isotope of uranium-233 as the way to determine the rate of the exchange, okay? I know that's a little bit too brief perhaps to go into a lot of detail. We'd have to make it clearer, but I gained a thorough knowledge of the use of radioactivity in working with Dr. Brubaker on this project. This research experience made me eligible and suitable and able to accept a research position at Oak Ridge National Laboratory, working there with other actinides like americium and curium, both in hot cell environments. In my 4th year at Oak Ridge, I received notification that my position was being eliminated because of the federal cutback in support of the national laboratories. That was very puzzling because I had a wife and 5 children and no commitment to go anywhere. And so after several unsuccessful attempts to get a position, that is, they just weren't readily available at that time in life, I contacted Dr. Brubaker to see if he had any insight into a position in industry or in university. And indeed he had. His suggestion was contact Barney Rosenberg. Barney Rosenberg was a young professor at Michigan State at that time, just a year or 2 here. Dr. Rosenberg was looking for someone who could carry out, to synthesize and label cisplatinum in the cisplatinum molecule with a radioactive nuclide that could help out in determining, one, its activity, its uptake, distribution, retention, and elimination from the mammalian system. Indeed, that was a fantastic opportunity. Anyhow, Dr. Rosenberg needed someone to do this work, and indeed I joined his group and spent approximately 3 years with him. The experience in Dr. Rosenberg's lab changed my life, both personal and professional. The synthesis and anti-tumor testing of numerous platinum complexes in collaboration with Michael J. Clear, who worked with me as well, in Dr. Rosenberg's lab led to the patenting of carboplatin, a very effective and successful FDA-approved anti-cancer drug used worldwide. Royalties from carboplatin have made it possible to promote Dr. Brubaker for this honor, and I am pleased that Dr. Brubaker is being honored in this way. I am also very happy Professor Tu Wong has the endowed professorship of Barnett Rosenberg Professorship. That's it. Thank you. I don't know either. I'm glad about this.
Yeah. Thank you, Jim. It's great to hear how that came about. I hadn't heard that story before. That concludes the formal part of the celebration today. We now invite everyone to continue the celebration in the post-ceremony reception, which will be right back there. Thank you very much.
We're looking forward to that.